Hello everyone. In this video I'm going to be talking through the process of translation. Now translation is the second stage of what we call protein synthesis. I've done a video on the first part, which is transcription, whereby we're producing what's called a messenger RNA molecule using DNA as a template. And in this process what we're going to do is we're going to use that messenger RNA and the information that it carries in order to make a polypeptide. So we're going to take that messenger RNA from the nucleus into the cytoplasm, where we're going to translate that base sequence of messenger RNA into the amino acid sequence of a polypeptide. Now the translation process incorporates about 20 different amino acids in the precise sequence dictated by 3 base codons, built from the alphabet of 4 bases. So what I'm saying here is that the messenger RNA that we produce from transcription is made up of lots of different bases, the letters A, U, C and G. And we read those bases in sets of 3. It's referred to as the triplet code. So each 3 bases of the messenger RNA dictate which amino acid goes where in the polypeptide chain. So I've got a picture here on the left of the screen that summarises translation. Now I'm going to expand on this picture. I'm just really using it as a kind of base thing to refer to. Now you can see what we've got here is the ribosome. First of all, we're going to imagine this entire process is happening in the cytoplasm, so that's key. We can see we've got the ribosome at the bottom here. Now the ribosome, just a few bits about the ribosomes are actually made of what's called ribosomal RNA. So that would be a little R with a capital RNA, and protein, and they actually have 2 subunits, a large subunit and a small. And it's not particularly obvious in this picture, but there are 2 subunits. And we can see we've got our messenger RNA at the ribosome here, and it's referred to as mRNA, especially in my other videos. That's just something to note there. And what we can see is that there's this thing called tRNA. Now the T stands for transfer. So I'll just make a note that tRNA is often actually written like this, and that T means transfer RNA. And you can see that there's an incoming tRNA in this picture bound to an amino acid at the top, this PHE. Usually the amino acids are abbreviated to the first 3 letters. So we've got, for example, PHE, ASP, LYS. They're all just shorthand versions of the amino acids they refer to. And we can see that these tRNAs seem to be coming towards this messenger RNA. Then we're getting, at this point here where I draw a star, some kind of bond. Now in this it's a peptide bond forming between amino acids. And then this tRNA dissociates and we've got an outgoing tRNA. So what we're going to do is talk in detail about this process and what is happening. Now the first point to mention is this thing about the tRNA. So I've said that in transcription you produce what's called messenger RNA, and I've done a separate video about the differences between RNA and DNA, RNA being ribonucleic acid and DNA being deoxyribonucleic acid, so a slightly different structure. This is tRNA, transfer RNA. Now over to the right hand side of the screen, I just want to expand a little bit more about what that tRNA or transfer RNA actually looks like, because it's drawn in the picture here as a block, a yellow block, but in real life what we have is something that looks a little bit like this. So I'll just draw it in place. It's not the best drawing, but it's just a little sketch. Now this, I suppose it's not particularly clear, but it would take too long to do this, but essentially running all the way along is this strand of transfer RNA, and each one of these squares that I've drawn, it's like this little red one here, the blue one here, green. So if you imagine that I did that all the way around, this is essentially one single strand of RNA nucleotides joined together. Now I say a single strand, there are points where the transfer RNA appears double-stranded, like here for example, where two parts of the strand seem to come together. Now what we actually have, just label this as our tRNA. What I'm doing here is just a sort of extra bit of notes about the transfer RNA specifically before we lead into the true details about translation. So you'll notice that this is clover shaped. We've got 3 loops in this transfer RNA molecule. This is loop 1. We've got loop 2 down here, and then we've got loop 3 over here. We have what's called the anticodon, at the bottom. Now again, this isn't drawn to scale, but the anticodon is a set of 3 bases that correspond to 3 from the messenger RNA. So 3 bases in the messenger RNA are called the codon, so a particular sequence of 3, and the corresponding complementary pairs to that are found on the anticodon. So this bit here is the what's called anticodon, and that's important for when we talk about how we're going to form this polypeptide chain. So for example, you can see over on the left hand side, if we refer back to the left picture, if we think of this bit here, GAU, that sequence of 3 on the messenger RNA, we can see that the anticodon is the CUA above it. Those 3 bases on the tRNA that correspond with that codon. So the 3 bases on the messenger RNA we refer to as the codon, and the corresponding bases we refer to as the anticodon, and you find the anticodon on loop 2 of the transfer RNA molecule. Now you'll notice that I've drawn one end of the tRNA at the top slightly longer than the other. Now this bit here, just where I draw a sort of black circle, that part there, that there is what's called the tRNA, or rather the amino acid for the tRNA binding site. So this bit here is our amino acid binding site, which is why I've drawn it slightly longer than the other. Now the amino acid is actually attached by a specific tRNA activating enzyme, and this requires energy. So then we've got some information about the tRNA. We've got a clover-shaped molecule. We've got a sequence of 3 unpaired bases called the anticodon. We've got one end attached to a specific amino acid, and we use a specific tRNA activating enzyme to do so. And we can see that one end is slightly extended beyond the other. So there is our basic structure of a tRNA molecule. So now it should hopefully make it easier to explain what's going on in this translation process. So the first stage of all of this is what's called initiation. So stage 1 is something called initiation. Now the small ribosomal subunit with a tRNA complementary to the start codon binds to what's called the 5' end of the messenger RNA, and it scans until a start codon, or AUG, is reached, and then the large subunit binds to the small subunit. Now in this picture essentially I've got the whole ribosome kind of just there over the messenger RNA, but in real life, as again just a bit further detail, that the small unit binds with a tRNA and then the large subunit comes into play. Now the tRNA complementary to the next codon along binds, and then translation starts. So what that then leads into is stage 2, and stage 2 is really what we're seeing in this picture. So stage 2 is the phase we call elongation. So all we have to do is initiate it. Now we've got elongation. Now, the large subunit of the ribosome advances. The tRNA molecules have, as I've said, specific anticodons for each of the 20 amino acids. And the first anticodon complementary base pairs to its corresponding codon, and then the second one joins, and so on. Now, the amino acids of adjacent tRNA molecules join together, forming a peptide bond, and the ribosome slides along 3 nucleotides. Now, the first tRNA then detaches from the ribosome, and the tRNA complementary to the next codon along binds, and just translation continues. So basically, if we were to put numbers to this, so this, if we were to say this particular tRNA here is number 1, so the tRNA comes down, and we can see we've got an anticodon of UUU. So uracil, uracil, and uracil, and that would therefore join with its complementary pairs of AAA on the messenger RNA molecule. It comes down, it joins, and we put into place this particular amino acid here, lysine. The next tRNA molecule comes, and then we can see the anticodon CUA binds with its complementary codon GAU on the mRNA, and we bring this second amino acid into place. So this is our number 2. And as I said, when the 2 tRNAs next to one another, we get this peptide bond forming between the 2 amino acids. And once that peptide bond has formed, tRNA number 1 can essentially then detach and come away, because the amino acids will be held together in what's in our essentially growing peptide chain. That's what our primary protein sequence is, essentially. It's just this growing polypeptide chain of amino acids, leaving just a tRNA molecule, and that tRNA will come off, as you can see on the left of the picture, and we can go and bind to the top of the tRNA another amino acid at this point here, this tRNA, or amino acid rather, binding site, as I've got on the right hand side of the screen. Now, what we have once we've had stage 2 of elongation is stage 3. Now, stage 3 essentially is what's called termination. Now, the ribosome translates the messenger RNA in what's called a 5 to 3 direction, 5' to 3' direction, until a stop codon is reached. Now, a stop codon, as I've mentioned in a previous video about the nature and structure of DNA, a stop codon will just kind of end this translating sequence. Otherwise, this chain will just continue growing indefinitely, and you'll make a non-functioning protein, essentially. So the polypeptide chain at that point that's formed is released from the ribosome. So this growing peptide chain you see here at the top of the screen, this will literally just detach. It will come away, and the ribosomal subunits will also dissociate from the messenger RNA and each other. So, as well as having this growing polypeptide chain, dissociating and coming away, this subunit of the ribosome will break off, this bit of the ribosome will break off, and the whole process will come to an end, or will terminate. Now, often more than one ribosome can be actively translating the same messenger RNA molecule at the same time, producing multiple peptide chains. Just one key term that some examples refer to, the complex of ribosomes formed is actually called a polysome. So just to repeat that, often more than one ribosome can be actively translating the same messenger RNA molecule, because clearly you're not going to just want to produce one single strand of peptide chain. It would be inefficient. So you'd have multiple translated versions of that, if you like. Now, the final bit is stage 4. Now, and some textbooks and websites don't really refer to stage 4, but I'm going to just for completeness sake. This one I like to call activation. So we've had initiation, elongation, termination, now activation. Some proteins need to be activated by a chemical called cyclic AMP. And it's written with a small c and then the capitals AMP, cyclic AMP, after translation to alter their 3D shape to be able to better fit their complementary molecules. So just a little bit extra there, that we need this activation of our polypeptide, of our protein that we've essentially made, by a molecule called cyclic AMP. And what it does is alter the 3D shape to better fit complementary molecules. And just a side note, that in prokaryotes, so we're talking about let's say bacteria, for example, eukaryotic being animal and plant. So in prokaryotic cells, as the DNA is loose within the cell, as in not in a nucleus, translation actually begins as soon as the messenger RNA has been made. Which implies in this there's a bit of a time delay, but as soon as the messenger RNA molecule has been made in a prokaryotic cell, translation instantly occurs. Okay, so then we have a little bit about protein synthesis, part 2, and the process of translation. Now it's a little bit more involved than transcription, I would say, but just in summary, so you have the ribosome, the messenger RNA molecule that's been transcribed, comes to the ribosome, and all of these transfer RNA molecules with amino acids attach them, come and bind to the messenger RNA. Essentially they bind in a set sequence, and they bring all these amino acids next to one another. Peptide chains form between those amino acids, and essentially what we get is this growing peptide chain. And once the peptide chain starts to form, the tRNA molecules dissociate from the mRNA and can go and pick up another amino acid essentially. And it all goes off complementary base pairing. Okay, hope all that helps.